Okay, welcome to chapter 3, the cellular level of organization. In this chapter, we're going to talk all about human cells. So this is going to cover the Principles of Anatomy and Physiology book by Tortora, sections 3.1 through 3.5. I'll make a separate video covering the rest of this chapter, so we just focus on the anatomy and the structural parts of our cell. And then we'll look at the protein synthesis and cell division, really the kind of functional physiology of our cell in a second lecture. So let's talk about what cells are. Cells are the most basic living, structural, and functional units of the body. So we are able to remove a cell from a patient and we can identify what type of cell that is and who that cell belongs to. We are going to look at the three main parts of the cell. The cell has three large regions. Then we're really going to go through the individual parts of each one of these as well. The first part is going to be our plasma membrane that separates the inside of our cell from the outside world, which is really important. Just like the walls of your house, we want to make sure that only certain things can get into our cell and certain things can get out. We need some things to stay inside and we need some things to stay outside. So we're going to go through how that works. We're also going to look at the cytoplasm. So the cytoplasm is everything inside of our cell. This includes a fluid, which we'll talk about, that's a liquid, and our organelles, which are small little compartments that have different jobs. And then finally, we will look at the nucleus that contains the genetic material of DNA. Here's a picture of the slide and all of the cell and all of the parts of the cell that we are going to go through together. You have a bit of a different picture from your textbook than I have in the PowerPoint, but it's showing you the same structures. This slide is a nice overview with all of the names of the parts of the cells, their general descriptions, and then a lot of people use an analogy to help study the parts of the cell. This slide gives you the example of the cell as a city. So it's going to compare each organelle and each part of that cell as a different part of a city. So you can kind of picture these really, really small microscopic things on a larger scale and kind of build their functions off of functions of things that you already know. So if that analogy works for you, this is a great slide to revisit after we finish our lecture. Okay, let's jump right into that plasma membrane. So again, the plasma membrane's job is to keep certain things inside of the cell and other things outside of the cell. To describe it structurally, we use something called the fluid mosaic model. Fluid meaning it must be movable. It is slightly rigid, and mosaic, meaning it has lots of different components, like a mosaic piece of artwork. So when we look at that cell membrane, we're going to see lots of pictures of this looking flat today. But realistically, I want you to picture like a basketball or a Rubik's Cube, some 3D circular or square structure that is actually going to be covered in our plasma membrane on all sides of it, not just the flat level that we're going to look at it at. So this fluid mosaic model is going to be really important when we're looking at the different pieces of that plasma membrane, especially on our next slide. Functionally, what that fluid mosaic model does is make a selectively permeable membrane, meaning we're going to keep certain things inside the cell and certain things outside the cell. So just like your house has walls to keep things in and things out, you also have doors and windows that are going to allow certain things to enter and exit at certain times. And so the cell kind of works the same way. We are going to see that selectively permeable piece means that certain things are going to be able to move in and out of our cells. So looking at the structural components of our plasma membrane, most of your plasma membrane is going to be composed of phospholipids. We talked about these in chapter 2, and they contained a phosphate head and two fatty acid tails. So a phospholipid is made of a phosphate and two fatty acids. Because of these structural components, this makes a phospholipid amphipathic, meaning one side is hydrophobic, it does not like water, those are those fatty acid tails, and one side is hydrophilic, the phosphate head, which does like to touch the water. So if you look at the picture on your slide or the picture I have in this PowerPoint, you can see our phospholipids actually make up a double layer membrane. So that's our phospholipid bilayer. The fatty acid tails are going to stick in and touch each other, while the phosphate heads will touch the outside of the cell and the inside of the cell where there is liquid or water in both places. So this phospholipid bilayer is really what helps maintain that selectively permeable membrane. Not too many things are going to be able to pass through that phospholipid bilayer directly, and they're going to need doors and windows just like in your house. Another component of our phospholipid bilayer, or our plasma membrane, that's kind of embedded along with those phospholipids are called glycolipids. Glyco for sugar, lipid for fat. Glycolipids work largely as a cell signaling molecule, meaning that they are going to be able to identify that cell to other cells. As your cells are doing their jobs in the body, they're going to run into other cells. Some of those cells' jobs are kind of to work as a security team and make sure that your body only has good cells in it, meaning your cells and not a pathogen like a bacterial cell. So these glycolipids help indicate that cell belongs to you and that cell's function or maybe location. We'll look at some specific glycolipids and glycoproteins as we go through certain units of our textbook and talk about different cells in different areas of the body. Glycoproteins also work this way. Glycoproteins are a sugar, and a protein component to them. These work very well for cell identification. We are also going to find some cholesterol embedded in our cell membrane. Cholesterol is a steroid lipid that we talked about in chapter 2. And cholesterol really helps maintain a rigid cell membrane. Some of our cells, we need to have a very distinct shape and maintain that shape despite many stresses, and to hold kind of that shape to do its job. Other cells need to be very flexible and be able to slightly change shape or modify their shape as they interact with different components of our body. So some cells may have lots of cholesterol in that cell membrane. Other cells may only have a little bit. So it's going to vary depending on where we're at in the body, how much cholesterol that cell has in its membrane. Finally, we see multiple types of proteins here. We see integral or transmembrane proteins. Transmembrane proteins span or cross the cell membrane. You're going to see a piece of them sticking on the outside of the cell and a piece on the inside of the cell, and they're going to cross that entire phospholipid bilayer. You can see some examples in this picture of our alpha helix protein, a channel protein, and then there they have the integral protein labeled for you. All three of those are integral or transmembrane proteins, meaning they go across or span the entire cell membrane. We also see some peripheral proteins in this picture. On the outside, there's this globular protein labeled right here. And on the inside, we see one labeled as a peripheral protein. Both of those are associated with the periphery or the edges of the cell, so we call them peripheral proteins. They are only going to be associated with one side of the cell or the other, the outside or the inside. They are not going to cross that entire cell membrane like your integral proteins. Looking at the types and functions of our membrane proteins, we see a lot of them on this slide. You're going to see many different types of cell membrane proteins depending on what that cell's job is. On your PowerPoint that's posted on your D2L, you can see the picture of all the different types of membrane proteins. The first one in that picture is called an ion channel. This helps ions, meaning charged particles, to be able to cross that cell membrane. Things with that positive and negative charge do not readily cross the cell membrane, and so they need a channel to do so. Ion channels work just kind of like a door with a bouncer on it, where you're going to show your positive or your negative charge, kind of like an ID badge, and then you're going to get let into the cell. So ion channels open and close regularly. They're going to allow lots of ions in and out of the cell. It's just they can't pass through that plasma membrane. The next one that you're going to see on your picture on your slide is a carrier protein. The carrier protein is a little bit more specific than the ion channel. It's going to have a molecule that's a specific size or shape, bind to it. It's going to close the opening to the outside world and then open into the cell, allowing just that molecule that's attached to the carrier to be transported into the cell. It can also work in the other direction, taking things from the inside of a cell to the outside world. The third one on our picture is our receptor. So receptors are embedded in that cell membrane, and they're kind of going to relay a signal. So the chemical or ligand is going to attach to the receptor on the outside, and it's going to cause an effect on the inside of the cell without that molecule ever entering our cell. So with receptors, rather than allowing things in and out of the cell, they're going to really just relay a message into the cell. We then see our enzyme. Enzymes are proteins that help speed up the rate of our chemical reactions that we looked at in chapter 2. So here we can see this cell has an enzyme embedded into its plasma membrane, and it's taking its substrate and forming its products right on the surface of that cell. We see this a lot in the digestive tract, where dietary molecules for you to actually be able to absorb those nutrients are very small. And if we would break them down into the size that we could absorb them before we were at the cell level, we would lose those little pieces. So right in your small intestine, those particles are going to be broken down into their smallest, most absorbable form right at the cell that's going to absorb them. That way we don't lose anything. Our next to last one in this picture is called a linker protein. This does exactly what it says. It links things from the inside to the outside, meaning it's going to attach different parts of the cell from the outside world to the inside. This usually helps provide strength and stability to our cell membrane. It may also help move or link two cells together. And then finally, those cell identity markers are glycoproteins that we talked about previously. Again, glycoproteins really help identify your cells as your cells to make your immune system know what it is looking for. So how do other things move in and out of the cell? How do things that don't need a protein get across that cell membrane? And this has a lot to do with something called a concentration gradient. In this picture, we see a chemical gradient, an electrical gradient, and then our electrochemical gradient. The chemical gradient is going to work off of the amount of chemicals or the amount of substrate on either side of the cell membrane. So we see these little red X's. There's about 7 of them inside the cell and 2 outside the cell. And the red arrow is showing you the direction that the chemical gradient would be flowing. So we're always going to go from high concentration to low concentration, meaning where we have more of something to where we have less of it. That's going to those little X's are going to try to move out of the cell and equalize the same amount on either side. In humans, that doesn't happen usually because things are always moving. But with our chemical gradient, we're just going to look at where are those chemical pieces flowing. Where is there more? They're always going to move over to the other side for less. With our electrical gradient, we are looking at electrical currents. And in the body, we are usually looking at ions. So you're going to have a driving force for our cations, our positively charged ions, and a force for our negatively charged ions. Here we see we have lots of negative charges in the cell and positive charges outside of the cell. That is normally how the human cell works. Most of the large negatively charged particles inside of a cell are actually unable to leave that cell. And most of the ions surrounding your cell are positively charged ions. So there's going to be a small electrical driving force for that negatively charged particle to push outside of the cell, and a larger positive force pushing into that cell based on trying to make each side of that membrane equal with positive and negative charges. Then finally, you can see a really nice picture of this on your slide from your PowerPoint, but we see our electrochemical gradient. this is what's actually happening in human cells. We are adding that chemical driving force with our electrical driving force to balance them out to an electrochemical driving force. You can see in the picture on your slide that there is a large chemical driving force pushing out because we have 6 potassiums inside of the cell and 3 outside of the cell. So those molecules, based on their chemical gradient, are going to want to move outside of the cell. We have a positive electrical force pushing in. And so we are going to see that electrochemical force be a balance between those two and actually pushing out of the cell. How this applies is when we are looking at how we are moving things across that cell membrane without those protein carriers. So now we're going to talk about that membrane transport. Let's look at how these cells do their job by moving things in and out without those with those chemical gradients and electrical gradients. So the first one we see is our passive transport. Passive transport does not require any energy, and it's going to move substances down their concentration gradient, again, from that high to low concentration. We're going to look at 2 types of passive transport, diffusion and osmosis. We can also have something called active transport that requires ATP or energy, and this moves substances against its concentration gradient. We're going to talk about primary and secondary types of active transport. And then finally, we're going to look at some vesicle transport. Vesicles are little bubbles or pouches or tiny sacs that move things in and out of the cell and that are composed of components of the cell membrane. We have 3 types of vesicle transport we're going to talk about: our receptor-mediated endocytosis, our phagocytosis, and our exocytosis. Here on this slide, we're going to talk about diffusion. Diffusion is the passive random mixing of molecules together. It's going to depend on mainly that steepness of the concentration gradient. So do we have 50 particles on one side of the cell and 2 on the other? That's going to move things pretty quickly across that concentration gradient to try to make them equal. Or if we have 5 particles on one side of the cell and 6 on the other, it is going to go a lot slower because that concentration gradient's a lot closer. Temperature is also going to affect the rate of diffusion. As temperature increases, so does the rate of things being able to diffuse. As things get colder, we will slow down that rate of diffusion. The size or the mass of the diffusing substance also can alter the rate of diffusion. So the smaller something is, the easier it will diffuse. The larger something is, the slower it's going to diffuse. And then finally, our diffusion distance. How far does that substance have to go? Is that cell membrane very thin and things can easily cross back and forth? Or is there something in the way? The thicker that cell membrane or the thicker that diffusion distance is, the wider it is, the slower that rate of diffusion will occur. So let's look at simple diffusion. Simple diffusion is the passive movement of things from high concentration to low concentration across that cell membrane. This involves nonpolar hydrophobic molecules most of the time because, again, they need to get through that phospholipid bilayer and those fatty acid tails. So things like our gases, oxygen and carbon dioxide, can do this. Anything that has a component of a fatty acid, a steroid, or fat-soluble vitamins are going to be able to do this. And things that are small and uncharged, like water and urea, will also be able to do this. And you can see on the picture on this slide, we are going to move from high concentration, where we have a lot of those little red particles on the outside of the cell, to an area of low concentration inside of the cell, until those particles reach what we call equilibrium, where they are equal on both sides. Equilibrium, again, is only going to happen very temporarily or not at all in human cells because things are constantly moving in and out. It's not once we get to equilibrium, we stop. Nothing is going to stop moving in a living human cell. It's just going to keep changing that gradient. Here we see our facilitated diffusion. Facilitated diffusion is also a passive process, meaning that it does not require any energy. And we are going to have two ways to do this. So in this picture, it looks almost like the picture that you have in your PowerPoint, but you can see diffusion. Those particles are just going to go from high to low concentration through the plasma membrane. Where facilitated diffusion, we are going to use one of those integral or transmembrane proteins for those little molecules to go from one side to the other. Our channel mediated facilitated diffusion uses a channel, like an ion channel, to allow those positive or negatively charged ions to travel across from one side of the cell membrane to the other. We also have carrier mediated facilitated diffusion, where a specific molecule is going to bind to that carrier. The carrier will then close to the outside world and open to the inside world, allowing those particles in. In this example, it could also move them out as well. So channel mediated is usually going to involve ions that are going to have a specific charge. Carriers are going to involve specific solutes or particles that have a special shape, and that's what's going to allow them to bind to that carrier and enter the cell. We also see osmosis. So osmosis is the diffusion of water. So when we are looking at osmosis, we are not looking at the movement of particles. We are looking at the movement of water to try to get that concentration gradient to be equal on both sides. So that's what you see in this picture here. You see a membrane that is semi-permeable, but the ions or the particles, the little red dots, cannot move. Only the water can move. So we see in the first part of this picture, on one side of that membrane, we have a low solute concentration. On the other side, we see a high solute concentration. The water is going to want to move from the side of low concentration to high concentration to try to dilute that side and make it equal concentrations on both sides of that membrane. And that's what you see have had happened here in our picture. The solutes, the particles, did not move. Only the water moved. And then we end up with an equal concentration on both sides. The volume is different, but the concentration of particles to water is the same. So osmosis is the movement of water from high concentration to low concentration to try to reach equilibrium when the solutes or particles can't move. This is extremely important when it comes to human red blood cells. If you're going to work in healthcare, you will probably either give your patient an IV or work with a patient who has an IV line already established. And we give that IV line usually to give fluids and different medications. What we normally give to our patients is called isotonic saline. Isotonic is going to allow things to move in and out of our cell equally. So the water is going to move in and out of the cell equally, allowing that cell to stay the same size and shape, not altering its volume, just providing some extra fluid. We can also see in this picture our hypertonic and hypotonic solutions. In a hypertonic solution, that solution is going to have more particles than the concentration of our red blood cells. So the water is going to want to move out of those red blood cells, causing that cell to shrink and leading to its possible death via what we call crenation. So it basically just shrivels up and dies because it doesn't have enough fluid inside of it. We can also use hypotonic solutions. That solution has less volume of solutes or particles in it than a human cell, and this is going to cause water to move out of that, or water to move into that cell, which could lead to the rupture of that cell via hemolysis, which is the lysis or rupture of a red blood cell. So when we are looking at our different IV fluids, it's really important that we always double check what we are going to give our patient and that we are giving them the right type of fluid, because we want to make sure that we are treating the correct condition. Again, most of the time it will be isotonic. There are very special, rare situations that we would use hyper or hypotonic solution. Jumping into our active transport now, our main difference here is this type of transport requires energy, and cellular energy in the form of an ATP molecule. So with active transport, we are normally transporting things against their concentration gradient or polar or charged solutes or particles. We can have a primary active transport where we are directly using that ATP. And you can see that with our antiporter in the picture on this slide. We are moving a potassium in and a sodium out. Moving particles in opposite directions is called an antiporter. That transporter there is directly using the yellow molecule of ATP that you can see in the picture. So that is an example of primary active transport. Secondary active transport relies on a process that already used ATP. So this is more like a step 2 in a step-by-step process where step 1 required ATP. So you can see the symporter in this picture, bringing the sodium and the glucose into the cell, is indirectly using that ATP. It needed the ATP from the antiporter to take and remove the sodium to the outside of the cell, so it could bring that sodium back into the cell in exchange or with a glucose. So primary active transport directly uses ATP. Secondary active transport indirectly uses ATP, meaning usually in 1 to 2 steps prior, we have utilized that ATP molecule. And finally, let's look at our vesicle transport. We have 3 types of vesicle transport. You can see the nice pictures on your slide. But the first picture shows you the receptor-mediated endocytosis. This is very specific. A specific molecule will bind to a receptor, and then it will be taken into the cell via the process of endocytosis. So part of that cell membrane will kind of pinch together into a little bubble and be taken into our cell. We can also have phagocytosis. That is the second type of vesicle transport you can see on your slide. Phagocytosis is basically cell eating. So the cell is going to stick out these little projections called pseudopods, and they make almost like a little mouth, and they are going to be able to engulf or eat up any type of little microbe or particle that that cell wants to ingest at the time. On the picture on your slide, you are seeing a cell that is ingesting a microbe, and it's going to digest it and break it down and make it not harmful to your patient anymore. So phagocytosis, we're just ingesting some type of large particle into our cell. And then finally, our pinocytosis is where we are taking smaller particles into that cell. So phagocytosis is kind of like cell eating. Pinocytosis is kind of like that cell drinking a smoothie. So it's going to ingest smaller particles. All of those occur within a vesicle or a little bubble or pouch that came from our plasma membrane. So that's why we call those vesicle transport. All right, next thing we are going to cover is the rest of our cell, our cytoplasm and our organelles. Inside of our cytoplasm, we're going to find 2 things: cytosol, which is the liquid or fluid portion of our cell, as well as our organelles, which are these little areas that have specific functions. The first organelle that we are going to talk about is our cytoskeleton molecule. Cytoskeletons are networks of protein filaments that help give your cell shape and move that cell around. You can see a picture of these on your slide, but we have our microfilaments. These help with a lot of movement and different mechanical actions. Our intermediate filaments, which help hold your organelles in place, kind of like you're hanging a picture on the wall in your house. And then our microtubules, which we really use with a special organelle called the centriole or centrosome for cell division. This is going to help with that cell division that we'll talk about in our next lecture. The next organelle we're going to talk about are our ribosomes. Ribosomes are where we make protein. These work like little protein factories. They are going to take messenger RNA, mRNA, and they are going to translate that into protein and make a large chain of amino acids that form a protein. These little ribosomes exist in 2 places in your body or in your cell. They are inside of the cytoplasm where they're just hanging out. Those ribosomes are there to make up proteins for that cell. So the ribosomes that are free in the cytoplasm will make up proteins for inside of that cell. They can also be attached to our rough endoplasmic reticulum, our RER, and those are going to make proteins that that cell is going to export or send out of that cell for other cells in the body. Our ribosomes are composed of RER, ribosomal RNA, and that is produced by our nucleolus, which we'll talk about in a minute inside of our nucleus. Here we can see that endoplasmic reticulum. These are a network of tube-like structures that are attached to your nucleus. The first part that's coming directly off of the nucleus is that rough ER, that RER that has our ribosomes on it. Again, that's there to make proteins that we are going to ship out of this cell. That RER is continuous with our smooth ER, our SER. The smooth ER has lots of enzymes, and it's able to detoxify and break down really harmful substances. It also is able to synthesize fatty acids and steroids, especially hormones. So we'll talk about that SER as we go through different parts of the body, like the liver and things like that, have lots of SER to help detoxify things. Kind of right next to that ER in our cell is our Golgi apparatus or Golgi complex. This Golgi complex really fits that analogy of the city, and it really fits the story of the post office. So what the Golgi is going to do is it is going to receive the proteins that the rough endoplasmic reticulum, that RER, made. And then it's going to prepare them to be shipped out of the cell. So it's going to package them up into a little vesicle usually. It's going to sort them for where they need to go. And then it is going to export them or ship them out of the cell. It is going to send all of those proteins out into little vesicles. They're going to merge with that cell membrane, and then they're going to be released outside of that cell to go wherever they need to go to do their job. While we are doing all of that, producing proteins and packaging and shipping things, we make a lot of debris. There's a lot of kind of trash, if you will, that builds up in our cell. And our lysosomes are really there to take care of that. Lysosomes are very bubble-like organelles that kind of look like perfect circles in that picture. And they are going to contain digestive enzymes that are able to break down lots of different substances of the body. Those lysosomes are going to fuse with something called a phagosome during phagocytosis, and they are going to break down whatever they've ingested into a less harmful or not harmful at all particles that it can then release out of that cell. So lysosomes really work kind of like a little garbage disposal. They're just going to break up whatever you put inside of them. On the same line as our lysosome, we have something called a peroxisome. Peroxisomes also are little bubbles with enzymes in them, and these peroxisomes contain enzymes called oxidases. These help with the oxidation and breakdown of fatty acids and amino acids. They are going to also detoxify different substances like alcohol and other things like that. We also find many of these in the liver. So peroxisomes are really going to help break down anything that it could damage your cell via the process of oxidation. And then finally, our proteasomes. Proteasomes work like a little recycling center. So they're still going to work like that lysosome and peroxisome, but they're going to break down your old proteins, and they're going to recycle those individual amino acids. This is really important because some of those amino acids can be hard to get from our diet. So we want to make sure that we're able to utilize them fully and just recycle them to make a new protein. That's a lot of work making all those proteins and cleaning everything up. So your cell's going to need a lot of energy to do that. So here's our mitochondria, the powerhouse of the cell. That mitochondria is going to make our ATP, our cell energy. And it has a specific double membrane structure that you can see in this picture. As tissues increase their energy demands, they will also be able to increase their amount of mitochondria because these little guys can just replicate themselves anytime that cell needs more energy. We'll go through the functions of the mitochondria more in AMP2 when we get to our metabolism lecture. And finally, let's look at our nucleus. The nucleus is mainly that dark staining center of our cell. It contains the DNA, which is all of that genetic information for that cell to be able to do its job. DNA cannot leave the nucleus and has to be copied to RNA first. So we'll talk about that in the second lecture for chapter 3 in a different video. That nucleus is protected by our double membrane called our nuclear envelope, which helps keep DNA in and almost everything else out. It does have some pores in that membrane, so RNA can enter and exit. Inside of the nucleus, we find the most darkly staining area. It's called the nucleoli or nucleolus, and this is where our ribosomes or that RNA are made. Table 3.2 in your textbook really goes through all of the organelles and their functions, so definitely check that out. And for the rest of chapter 3, see the following video.